Amino acids, peptides, and proteins. Chapter 25. Objectives. 1. Introduction to amino acids, peptides, and proteins. 2. Amino acids. 3. Peptides. 4. Proteins. Your goals. By the end of the chapter, you should be able to draw the structures of amino acids, understand the role of the side chains, predict amino acid reactions to form peptides, understand the 4 categories of protein structures, identify some functions of proteins. 1. Introduction to amino acids, peptides, and proteins. It might be necessary to review the following topics to prepare for the material in this chapter. Designating RS configurations of a chiral center, structure and reactivity of carboxylic acids, nucleophilic acyl substitution, properties of amines. Introduction: amino acids. An amino acid contains an amino group and a carboxyl group. Alpha amino acids contain an amino and carboxyl group that are separated by one carbon, the alpha carbon. All alpha amino acids, except for glycine, are chiral. The alpha carbon is the chiral center. Introduction to peptides. These are relatively short amino acid chains, usually containing less than 40 or 50 amino acids. The prefixes di, tri, tetra are used to describe a peptide of a specific length. Polypeptide refers to chains that have fewer than 40 to 50 amino acids. Peptide bonds are amide linkages that connect the amino acids. Introduction to proteins. Proteins are amino acid polymers. These are larger chains than peptides. The term protein itself implies a biological function. 2. Amino acids. Each amino acid has a name, a 3-letter and a 1-letter abbreviation code. 20 alpha amino acids occur naturally. They differ in the identity of their side chains or their R groups. Natural amino acids are called L amino acids. The stereochemistry resembles L sugars. Our bodies can make 10 of the 20 amino acids we need. We must get the other 10 amino acids from our diet. Meat eaters can get all of their essential proteins from meat, fish, milk, and eggs. No single plant source contains all the essential amino acids. Vegetarians and vegans need a combination of grains, beans, and nuts. Let's consider the side chains. Amino acids with nonpolar side chains are shown here. They're including their name, their structure, and their abbreviation, both their 3-letter and 1-letter abbreviations. Polar side chains. These are the amino acids with polar side chains. They're given their names, their structure, their 3-letter and 1-letter abbreviation. Acidic and basic side chains. These are amino acids that either have a basic side chain or an acidic side chain. Again, note their name, structures, and abbreviations. Amino acids not found in protein. Some naturally occurring amino acids that are not found in protein include neurotransmitters and hormones. Acid-base properties. Under acidic conditions, which means a low pH, both amino and carboxyl groups are protonated, and each one of these groups will have a unique pKa value. We'll have pKa1, that's going to be for the carboxyl group, and pKa2, which will correspond to the amino group. pKa tables. The carboxylic acid is more acidic, than the ammonium ion. Acidic and basic side chains will also have their own pKa values. So notice we're given the pKa values here of the carboxylic acid, of the ammonium, and then if there's a side chain that's acidic or basic, we also have its pKa value. The pKa tables are continued here, where we have the amino acid, the pKa of the carboxylic acid of that amino acid, the pKa of the ammonia, and then the side chain if it happens in this case to be basic. pKa values example. The carboxyl group of alanine has a pKa value of 2.34. When the pH is equal to 2.34, the amount of protonated carboxylic acid and deprotonated carboxylate aniline will be equal. At pHs that are higher than 2.34, more alanine molecules will be deprotonated and vice versa. At physiological pH, the structure of an alpha amino acid is zwitterionic. Physiological pH is higher than the carboxylic acid pKa and lower than the ammonium pKa. So both charges will be present, the positive and the negative. Amphoteric. At physiological pH, amino acids are amphoteric and functions as bases and acids. When treated with an acid, the anion will react and then form the carboxylic acid. When treated with the base, the cation part will react and then will result in water and the amine. Isoelectric point, Pi. The pH at which the Zwitter-ion concentration is the highest is the isoelectric point. For an amino acid without an acidic or basic side group, we simply take the average of the two pKa values. For other amino acids, average the pKa value of the side group with the matching amino or acid group. For acidic amino acids, average pKa value of the side chain and the COOH group. For basic amino acids, we're going to take the average of the pKa value of the side chain and the ammonium group. Electrophoresis. Electrophoresis is used to separate amines by exploiting their Pi values. When the Pi is greater than the pH of the solution, amino acid is going to be cationic. When it's the other way around, the amino acid is anionic. If two amino acids have similar Pi values, the larger amino acid moves more slowly. Cations will move towards the cathode, anions will move towards the anode. Electrophoresis is an analytical skill technique. It isn't used to separate large amounts or quantities. Amino acids are generally colorless, but their presence can oftentimes be verified with an anhydride color indicator. Usually that will result in a purple colored product. Amino acid synthesis. Alpha haloacids. The Hall-Volhard-Zelinsky reaction is used to functionalize the alpha carbon of a carboxylic acid. Subsequent SN2 reaction with ammonia yields an alpha amino acid. Amidomalate synthesis. Like the malonic ester synthesis, Rx provides the side chain for the amino acid, and here's an example of the synthesis of phenylalanine. The Strecker synthesis. Amino acid synthesis via the Strecker synthesis. This is a two-step synthesis from an aldehyde. The side chain of the aldehyde becomes the side chain of the resulting amino acid. Strecker mechanism. This is the mechanism for the alpha aminonitrile formation. In the first step, there's nucleophilic attack. Ammonia attacks the carbonyl carbon. The pi electrons are bumped up. Then there is a proton transfer, another proton transfer, removal of a proton from HCl, which will then result in the loss of the leaving group water. This will undergo nucleophilic attack by cyanide, And then from this point, we can see that we end up with the formation of the carboxylic acid, recalling section 20.13 from the textbook. Enantioselectivity. Syntheses covered so far provide racemic mixtures. A desired enantiomer may be obtained in one of two ways. The first way is resolution of a racemic mixture or the separation of enantiomers, or asymmetric synthesis that will result in the production of only one enantiomer. It is clearly more efficient to synthesize the desired enantiomer as opposed to fishing it out from a racemic mixture. Enantioselective synthesis of amino acids. One way that this can be done is through asymmetric hydrogenation. Many chiral catalysts consist of ruthenium complex to a chiral ligand. The following example will provide D-phenylalanine with 99% EE. Note that the key here is the chiral catalyst. 3. Peptides. Consider a dipeptide. Two different dipeptides are possible, consisting of alanine and glycine. Glutathione is an example of a tripeptide. It isn't enough to join these three amino acids to make glutathione. They must be assembled in the correct order from end to end. Terminus nomenclature. Peptides have an N-terminus and a C-terminus. The N-terminus is where we have the amine. The C-terminus is the carboxylic acid. We always write the N-terminus on the left-hand side, and three-letter abbreviations can be used to indicate the sequence of amino acids from N-terminus to C-terminus. Rotamers. Recall that peptide bonds are amide bonds. The C-N bond has a partial double bond character which restricts rotation. As a result, the C-N bond exists as either the S-trans or the S-cis-trans rotamer. Peptide bonds of the peptide backbone prefer the S-trans conformation. Therefore, these bonds experience restricted rotation. The other backbone bonds can freely rotate. A variety of peptide conformations are therefore possible. Disulfide bridges. Formation of a disulfide bridge is critical to the structure and function of many proteins. Cysteine residues in a protein or two separate proteins can connect through a disulfide bridge or an S-S bond. Such linkages greatly affect the 3D structure and function of many proteins. Here is an example of having a disulfide bridge within a chain, or two different chains are attached via disulfide bond. Some biologically important peptides are going to be shown here below. Some function in the brain in control of pain. Some function as hormones that dilate blood vessels and act as anti-inflammatory agents. Others cause reabsorption of water by the kidneys and controls blood pressure. And others induce labor and stimulate milk production. Sequencing peptides via the Edman degradation. Removing an amino acid from the N-terminus of a peptide will result in a PTH derivative. The PTH derivative is analyzed to identify the amino acid. This process can be automated and is repeated until all the amino acids in a peptide have been identified. The mechanism for the Edman degradation is shown here below. Make sure that we understand that Edman degradation allows us to identify the N-terminus amino acid. Sequencing peptides. Enzymatic cleavage. Larger peptides, those that are longer than 50 amino acids, are broken down into smaller chains with peptide diases for sequencing. A given peptidase will selectively hydrolyze specific peptide bonds. Trypsin cleaves a peptide bond on the C-terminus of arginine and lysine. So we would get information then about the connections between lysine and arginine. Chymotrypsin catalyzes the hydrolysis of the carboxyl end of aromatic amino acids, phenylalanine, tyrosine, and tryptophan. The smaller fragments are then sequenced by Edman degradation. Synthesis of peptides. To form a peptide bond, the N-terminus of one molecule must be joined to the C-terminus of another molecule. DCC is a popular coupling agent used for that purpose. The mechanism is here, but just know that this is a method to synthesize peptides. Protecting groups. The coupling of two amino acids potentially will yield 4 different dipeptides. To make a desired dipeptide, protecting groups are needed. The N-terminus of one amino acid and the C-terminus of another amino acid must be protected. If they're protected, then only the ends that we want them to react will react. The protected amino acids are then coupled, followed by deprotection to yield the desired dipeptide. Types of protecting groups. Carbamate. An amino group can be protected as a carbamate. Tert-butoxycarbonyl, or BOC, is a popular carbamate protecting group. The mechanism of adding the group is shown here. The group is removed easily under acidic conditions using an acid like trifluoroacetic acid. There are other protecting groups. We can protect an amine using an FMOC group. The mechanism of installing the group is very similar to the previous protecting group BOC, and it's removed in this case under basic conditions. Here is an example of removing the group under basic conditions. Types of protecting groups. Esters. The carboxyl group of an amino acid may be protected with an ester. Methyl and benzyl esters are the most popular. Hydrolysis is generally used to cleave or deprotect the ester. Benzyl esters are popular because they can be deprotonated without using acidic or basic conditions. Hydrogenation can remove a benzyl group. Preparing peptides. Preparing a dipeptide. Peptide linkage is accomplished by 1. Protecting amino acids, 2. Coupling, and then 3. Deprotecting. We saw step 1, protecting, step 2, coupling, step 3, deprotecting. The method of protection, coupling, and deprotection is not practical for forming larger peptides. For every amino acid added to a peptide, 3 reactions and 3 purifications will be necessary. Larger peptides. The Merrifield synthesis is a superior method, reducing the number of synthetic steps significantly. The peptide is tethered to an insoluble polymer. This is called solid-phase peptide synthesis, or SPPS. The Merrifield synthesis. The C-terminus of an N-protected amino acid is attached to the surface of a polymer. It attaches via an SN2 reaction. The N-terminus is deprotected, and the next BOC-protected amino acid is coupled to the chain. Once the peptide is finished, it is cleaved from the resin using hydrofluoric acid, HF. Benefits of solid-phase peptide synthesis. The real advantage to SPPS is the ease of purification. The polymer beads are dropped in solution when the reaction is done. They are collected by filtration and plopped into the next reaction and can be used again. The process is now completely automated. Pre-made solutions of amino acids and DCC can be pumped in and out of a reaction vessel leaving the polymer beads in the reaction vessel throughout. For this work, Merrifield won the Nobel Prize in 1984. 4. Proteins. 4 levels of protein structure, primary, secondary, tertiary, and sometimes quaternary, are needed to describe the 3D shape of a protein. Primary structure. The primary structure is just the amino acid sequence. Secondary structure. The secondary structure refers to the 3D shape of localized regions of a protein. These are largely dictated by hydrogen bonding interactions along the peptide backbone. There are 2 common arrangements, the alpha helix and the beta pleated sheet. Tertiary and quaternary structures. The tertiary structure of a protein refers to the 3D shape of the entire peptide chain. The shape will change depending on the solvent and the environment. Polar environment, proteins will adopt conformations that will put polar groups around the exterior. In non-polar environments, polar groups will be in the interior. The quaternary structure results when a multiple set of peptide chains aggregate to form a protein, or we have 2 or more chains needed for it to be functional. Hemoglobin is an example of one. It is composed of 4 peptide chains. Denaturation. Intramolecular forces are holding the protein together, and if those are disrupted by temperature, pH, or anything else, that will result in the denaturation of that protein, and it can no longer function. Protein functions. There are proteins that function as structure proteins. These are also known as fibrous proteins found in hair, skin, and fingernails. They provide structural rigidity by bundling together. Several polypeptide chains of alpha-keratin bundle coil together in hair. Enzymes. Enzymes are globular proteins. Enzymes catalyze biological reactions by stabilizing transition states and/or reactive conformations. The lower the energy of the transition state, the faster the reaction. Some function as transport proteins. Transport proteins are used to transport molecules from one location to another. Hemoglobin is the classic example of a transport protein. It contains prosthetic groups, or heme, that bind the O2 molecules for transport.